Hello everyone, my name is Chloe Wiseman and I'm a distance learner from Central Massachusetts, and my presentation is on population genetics. So I wanted to begin by going over the principles of population genetics. It is a field in biology that deals with the genetic content and variation of biological populations and how they change over time. So this discipline really binds Mendelian genetics with Darwin's theory of evolution. It's considered a cornerstone in modern evolutionary theory. And we kind of take it for granted now that genetic changes underscore evolution. But back when Darwin wrote Origins, he did not understand how traits were passed onto their offspring. So it's unfortunate that these two men who lived and worked at the same time never got to learn how their work connected. So population genetics is kind of like the mathematical glue that binds Mendelian genetics with evolution. So it aims on shedding light on the evolutionary process by evaluating changes in allele frequency. And it does this with statistical methods to predict genotype frequencies that can then be compared to the empirical data of phenotype. And from that, we can try to understand the forces that change genetic makeups in populations. So a major breakthrough in this field was in 1908 by English mathematician G.H. Hardy and German physician W. Weinberg. And they independently came up with an equation known as the Hardy-Weinberg principle. And it's a concept that shows how Mendelian genetics can be applied to large populations. So it's an idea that the alleles will reach a genetic equilibrium in a stable population. And you can add the frequencies of these alleles. So you can add the frequency of allele A, or big A, denoted by P, and the frequency of allele little a, denoted by Q, and the sum of these two frequencies will equal 1. So since humans and many other species are haploid, we can square this, and we can say that P plus Q squared equals 1, and extrapolate that out to P squared plus 2PQ plus Q squared equals 1. And this is useful to help us determine allele frequency and the amount of heterozygotes in a population using phenotypic traits that are governed by two alleles. So just as an example, you can say that the rate of babies born in Ghana that have the sickle cell disease is 2%. So we can use the Hardy-Weinberg principle to estimate how many people are carriers for sickle cell in Ghana. So we'll take the 2%, that's the recessive small a genotype. So that would be that would be Q squared. So we square so you take square root of 0.02, which equals 0.14. That's our frequency of the sickle cell allele. And we use this to find P. So 1 minus 0.14 is 0.86. So that's the frequency of all the other, quote, normal alleles. And we can plug Q and P into the Hardy-Weinberg principle to find our big A, little a frequency, which is about 24% as an estimate. But this principle comes with one big giant caveat. It is based on a population in equilibrium, which assumes that there is random mating, no mutation, no migration, and no natural selection. So you might wonder, what's the use of this? So it's useful in evaluating the equilibrium of genotype frequencies. So a population whose genotype frequency does not remain in equilibrium from one generation to the next generation is considered to be in disequilibrium. And therefore, you can assume that there is one or more evolutionary forces that are altering the genetic variants in the population. So these modes of evolutionary forces and population genetics are defined as natural selection, which we're familiar with, that makes one allele more or less fit for survival than the other. It's usually often environmental, can be infectious, climate, food supply. We have random mutation, mistakes in DNA transcription, which increase variation. Genetic drift is interesting. It's a random events that alter the expected offspring outcome that result in a higher frequency in one allele to the other, but this random event is not related to fitness. So, for example, it could be a compound of one kinship that is wiped out by a fire, and thus reducing the allele variance that they would offer to their population. So it affects smaller populations in a more dramatic way. And over time, these random events will eventually approach a population to a homozygous allele, since the random events will approach, will either like eliminate or become, have caused an allele to become fixed within the population, thus creating more homozygous genotypes. And genetic drift is also responsible for the divergence of subpopulations, because random events that occur to different subpopulations that are separated by distance, obviously they're going to be different for each group. So when we say random events, it's not to mean that we don't know what those events are. It means that they're random and that they're not related to fitness. And then lastly, we have gene flow, which refers to immigration and emigration, which again affects smaller populations in a more pronounced way. And finally, non-random mating is a force of evolution, which doesn't always change the frequency in alleles, but it refers to that certain traits are selected for one reason or the other to produce more offspring, but that it's not related to fitness. Okay, so how does all of this relate to blood banking, and why does it matter? So in a nutshell, it allows us to deliver safe blood products in a timely manner. So population geneticist research has allowed us to establish antigen frequencies in the populations that we serve. And as you know, it's a useful tool in selecting antigen compatible products for sensitized recipients. So the antigens vary within the population we serve, and we need to be able to identify that and use this information to calculate how many units we might need to screen in order to find a compatible product. So just a quick review for the final exam and the BOC. How would you know how many products to screen to find a compatible unit? So say we have a recipient who's immunized to antigens Z, Y, and X. So first we're going to determine the frequency of the antigen negative persons in the population by subtracting the known antigen frequency from 1. Next we will multiply all the antigen negative frequencies to determine the percent of persons that are negative for all 3 antigens. And then finally we set up a proportion problem to determine how many units to screen in order to find the number of units requested. So as a nice review for the exam, we can see some common antigen frequencies among populations. You can see here that the A phenotype is common among the white population. O phenotype is pretty prevalent amongst all white, black, and Asian. The D negative phenotype is more prevalent in the white population than Black and Asian. We go down and see high frequency antigens, little k, and Lutheran B. And then as we've discussed in class, the Duffy phenotype, where we have AB positive with 40% in the white population, but AB negative, very rare in the white population and 67% in the Black population. And as we've gone over, this is a defense mechanism against Plasmodium vivax. So in doing this project, I was very interested to see why there are so many polymorphisms within the human species on the RBC. And you can, as from looking at the previous chart, you know that blood groups are found to be population specific and can be used as markers for ethnicity. Some examples of this are the Diego antigen that we went over in class. It has a high prevalence in the Japanese population, but it's also been used to track the migration of East Asian populations to the Americas. And it has a rare occurrence in actually Southeast Poland, which denotes the reach of the Mongolian invasions in recent historic times. You can see that the JSA antigen belongs almost exclusively to people from African descent. The KID phenotype, AB positive, occurs frequently in Asian and Caucasian populations. And as we've gone over, the Duffy system varies across the world, but the AB negative is most often seen in Black populations because of its advantage against malaria. So when Landsteiner set out in almost 120 years ago, discovering the ABO system, he was testing to see if the differences between species could also be found within species. And this turned out to be the start of the study of genetic frequency and variation within different human populations. His early work, since Lancaster's early work, population geneticists have often used blood bank antigens to trace early migration patterns before the molecular revolution allowed us to study the alleles directly. And so from their studies, one can overlay the distribution of blood group frequencies on a world map and see the effects of genetic flow, of migration, natural selection, and genetic drift that they've had on human populations. So I wanted to illuminate three kind of interesting examples of this. The first is the D negative frequency in Europe. So it's most prevalent in white populations, as we saw in the chart, 15%. And the loss of RH-D protein doesn't seem to have too many negative effects in terms that the CE protein can kind of pick up the slack for the lack of D. It's theorized to facilitate the assembly of band 3 proteins and the band 3 protein gas transport complex. But there is a negative selection pressure against Rh negative alleles because of the hemolytic disease of the newborn and fetus and newborn. So, you know, it begs the question, why are Rhd negative alleles still so prevalent in Europe? And the answer to this is genetic flow, or also called the founder effect. And it's hypothesized that a small number of ancestors who survived the last ice age, about 10 to 15,000 years ago, had Rhd negative allele that was conserved. And that's why you see this allele so prevalent in Europe today. Another interesting case is for the O phenotype. So the O phenotype, as we know, is a morphism from the AB transferase gene, and analysis estimates that the AB transferase gene appeared after the separation of fish and amphibian lineages, so it's quite an ancient gene that we've conserved throughout evolution. And the AB glycoproteins seem to offer certain protections against infectious agents, because they're not only on the RBC surface, but also in the gastrointestinal tract. So, for example, group O patients have a greater likelihood of severe infection of cholera, and it's suggested that there's the reason for a low prevalence of type O people and a high prevalence of type B people in Bangladesh is directly related to the natural selection from cholera. And another example is actually recently from 1996 in Scotland. There was an outbreak of E. coli 0157, and this disproportionately killed individuals of type O, making 87.5% of the deaths. So there seems to be some advantage for being type AB. So why is type O actually so prevalent? So this is theorized that the type O polymorphism emerged in Africa before the Great Migration out of Africa about 70,000 years ago. And the type O polymorphism does have a couple of advantages. It has a greater propensity to clot from excess von Willebrand factor and factor 8. This was mentioned as an antidote in previous lectures. So this might have been advantageous for our ancestors to be able to clot faster. And there's also been studies that show that type O blood has reduced resetting of RBCs infected with Plasmodium falciparum compared to non-type O. So finally, I have another example of how infectious diseases have put an evolutionary process on our RBC antigens. So in other species, the red blood cells play a big role in immunity. For example, in birds and amphibians and fish, they have nucleated RBCs, and they upregulate viral response genes, produce chemokine-like factors, and even sequester pathogens. And although our RBCs are enucleated, we have conserved some of that immune function. So going back to Duffy that we are familiar with, they have been conserved throughout evolution, theorized because they play a role in chemokine regulation. They have the ability to soak up excess chemokine when necessary, and this can damper down the neutrophil signaling, and so that the immune doesn't go in overdrive. And it can also release chemokines under certain tissue conditions. However, as we've discussed, P. vivax exploits this antigen as an entryway into the erythrocyte. This is why we see a variation in Duffy alleles in malaria endemic locations. But P. falciparum uses glycoprotein A, B, and C to enter the RBC, yet these proteins remain one of the most common proteins on the RBC surface, so they haven't really experienced the same evolutionary pressure as Duffy did, which is strange. So not only do these proteins provide a negative charge necessary to maintain the zeta potential that prevents RBC agglutination, it's been theorized that these glycoproteins act as a pathogen decoy receptor, and they suck in different pathogens such as influenza and mycoplasma and E. coli, and they can bring it away from tissue and into the spleen to be phagocyzed. Hence why it maybe serves as a valuable conservation. So in conclusion, it seems that there's a combination of natural selection against infectious diseases genetic drift, and founder effects in small populations that explain the allele frequencies that we see today. It's important for the future because we are not a static species, and these evolutionary forces are still at play. With the era of globalization, our species is undergoing pockets of increased heterozygosity from genetic flow. And for mixed populations, it's always important to ensure that all ethnic groups are adequately covered because the frequency of alleles may differ between groups. First generation heterozygous people are a special case because they will have markers for two groups. Thus, they may be restricted number of suitable donors for them. And we're excited, and thank you so much for listening, and have a good day.